Yeah, hi guys. Welcome back to my channel. Okay, this video is particularly for STPM Sem 2 students. STPM Sem 2 students who are about to take their Sem 2 exam in 3 weeks time, 2 to 3 weeks time. All right. There will the first two things before we go through if you read the title, it's a topical analysis. It's a topical analysis. Okay, for your upcoming Sem 2 exam. Okay, two things first. Guys, two things first. For those of you who contacted me with regards to my Sem 3 notes, because after you finish Sem 2 with Sem 3, it's now ready in my Shopee. It's now ready in my Shopee. So as usual, similar to Sem 1 and 2, is well hacked, photostated properly, and you get all the topics one shot. Okay, for those of you who ask about my Sem 3 notes, I just updated it. Yeah. Okay. So that's the first thing. Now, second thing, which is a lot more crucial. At this point in time, technically, technically, today is 10th of August. Okay, today is 10th of August. So you have about 2 weeks. 2 weeks. Okay, 2 weeks to your PA2 Bio 2, then Chemistry 2 and Math 2. I just told my Sem 2 students this about all in about I think about 60 of them. I told my Sem 2 students, about 60 of them, never before, to my knowledge guys, to my knowledge, never before there has been such a comfortable gap for STPM students to do revision before exam. Typically, it'll be within the week. Okay? You have PA2 Math 2. Yeah, PA2, not normally, if let's say example, Physics 2 is here. Of course, if you're taking Bio, you won't be sitting for Physics. I mean, 99% of you. How many students take 5 subjects nowadays? Last time, my time also, I took 4. Guys, I took 4 subjects only. So did anyone your time take 5 subjects? No. No. Bio class taking physics or physics students taking bio subject. So normally it'll be the usual schedule whereby Monday is Physics 2, PA Math, Bio, and Chemistry. This is how it normally is. So guys, for the first time ever, again, to my knowledge, guys, as far as I can remember, I started teaching ever since the very first semester system came out. Even before the semester came out, I already started teaching. Ever since, never before there's been such a big gap for students to do revision. So what I'm trying to tell you guys is, the message that I'm trying to get across is, take advantage. Make full use of the time given. Make full use of the time given. Yeah, you have an age there. Okay, with that said, guys, we are around here now. Okay, if you're watching this the very day I uploaded this video, we are here today, 10th of August. Okay, 10th of August. Please do active revision. For bio, please do active revision. Now, most students around this point in time, if you are still reading a lot, it's not going to help you much because you are doing the same thing that you have been doing the very first time you learn in class or you learn by yourself or you learn in tuition. No difference, guys. There's no improvement. Kali pertama baca, kali terakhir sebelum masuk Dewan Peperiksaan pun baca. Where's the improvement? Yeah, 哪里有进步? So you have to test yourself a bit more. Do active revision. Oh, can I explain menstrual cycle? Can I explain menstrual cycle? Can I explain? Urine formation, yeah, or structure of liver. Also, liver there are four lobes. Okay, left, right, quadrate, caudate. Then they are made up of lobules. From there, you are able to elaborate the structures in the lobule. So if you are able to do that, guys, you are doing active revision because you will get stuck. Sometimes we procrastinate, guys. I know because I was a STPM student before. Kita tak nak buat sebegini sebab ia menakutkan kita. It scares us. No, not only you guys, but all the students, I would say, whether be it Form 4, Form 5, of course, Form 6 is worse lah. I shall not lie. Form 6 is worse lah. Okay, so you have to test yourself. From there, oh, sir, urine formation. I know there's ultrafiltration, selective reabsorption, active secretion. Yeah. Ultrafiltration, blood entering glomerulus is rich in waste substances. Diameter afferent bigger than efferent. Therefore, high hydrostatic pressure. Then I forgot whatever happened next. Itulah kelemahan Anda. You can't say it now. You can't write it out in exam. As simple as that. That's where your weakness is. You get stuck now, you get stuck in exam. Okay? So guys, number one, do active revision. I bet after this, you kind of get what I mean. Number two, guys, if you watch the session that I had with Huilin, the chit-chat session, whereby she shared how she managed her time, how she studied. Past year questions are very important. Now, in my class, I just finished my last class like two days ago, Monday. 2 days ago, I just finished. They have all the questions. My students have all the questions. We did 2005 to 2010. I don't really have 11 and 12 lah. So I gave them 2013 all the way to 2019. So guys, make sure you try past year question. If you have tried all these, hey guys, don't forget there's also ulangan. There's also ulangan. If you have tried all this, you should be quite steady. You should be quite steady. Okay, so I don't have that much time to try because it's already 10. Or if you're watching this even later, you have even lesser time, scan through the question. You don't have the luxury of time to attempt question by question. Objective takes up time, no joke. So just scan through the question, go through the answer, just to check your understanding. And even that, guys, don't go and do this 1, 2, 3, 4, 5 days before your bio paper. The time to slowly try question, slowly digest it, yeah, taking the facts, get to know the application is long gone. You should have done it 1 month ago. Okay, so this is where it leads me to my last point, guys. Avoid complicated subject matters. Don't suddenly out of nowhere you get the burst of motivation, then try all sorts of questions from, I don't know, the last 20, 30 years. A few days before your bio paper, you will kill your own marks. You will kill your own scores because, as you can see here, confidence matters. Guys, confidence matters. Because it is common sense that the more questions you try, the more you realize you do not know. Don't you think it affects you more adversely in exam? So again, I want to emphasize, all this should have been done the past 1 month ago. Okay, 2 months ago, should have done it then. Should have done it then. Okay, so guys, yeah, just a small tip. Now let's look at topically what should we. Of course, whatever problems you face here, the next 1 week, try to clear it in stages. The last 1 week before your paper. The last 1 week before your paper, that's where you really revise through the topics. Your confidence should be really there. That's why whatever hard questions you want to have a go at it, do it now, guys. Now, today, this very moment. Okay. And this applies to other subjects, chemistry especially. I need to emphasize chemistry especially. Yeah. I was an STPM student. Guys, I know what you face. I know what you face. What we took back then in STPM is longer and tougher than now. If you don't believe me, ask your teachers. Ask your teachers. Bio last time, my time was 25 topics. I still remember. We studied all one shot. We entered the exam hall, take and habis, no ulangan. Now you have 19 in 3 semesters, and every topic, not every topic, quite a number of topics, they shrink a little bit. Okay. Okay, now moving to the next thing. I think I did this for Sem 1, my YouTube. I think so. Anyway, so guys, this will be this handout is in the Google is in my Google Drive, whereby I'll share a link below. You can click on the link to get this analysis. And if you realize in Popular, they don't print any more of past year questions because of your juniors. Okay. I'm not asking you to blame your juniors, but if you want to, go ahead lah. Why your juniors? Because they are the first batch of KSSM. Ever since KSSM was implemented in lower forms, every publisher in Malaysia was shaky, very shaky. Okay, I'm with one publisher myself, but then it's for Form 4, Form 5 lah. Yeah, I'm not attached to any publishers in Form 6. I was offered lah, but I couldn't commit the time. Anyway, the reason why there's no more past year paper is because KSSM, your juniors, KSSM Form 5, first batch coming up to Form 6, everyone was expecting, I mean, most students and teachers were expecting a change in syllabus. But I think that's not going to happen. I started my Sem 1 class. I told I think about 70, 80 of them. I told them you'll remain lah. Definitely yours will remain. You don't have to worry. Just focus and study. Because of the degree of toughness in KSSM, they hike up. As let's say STPM is here. Last time I would say SPM is here. Now because SPM is tougher, so the degree of toughness that KSSM students have to overcome is much smaller compared to you guys, KBSM, last batch, Form 5 in SPM. Okay, now guys, I have two things here for you. Number one is actual paper analysis, then repeat, okay? And also repeat. Does repeat matter in your actual upcoming STPM? Not really lah, only a small margin. Not really, guys, not really. So I won't be basing it much on your repeat. Instead, it's mostly I'll base it on your actual. Because certain things you can check out yourself. Certain things that came out in actual, they adjusted a little bit for the repeat. Why? Because because why? Same batch of students, they don't want them to deviate too far in terms of the standard of questions. Faham tak? In fact, there were 1 or 2 years, the actual is the repeat. So how come actual and repeat same? Is it an error? Is correction needed? No, it's because they adjusted to a point whereby actual and repeat they took at the same time. Maksudnya menduduki set yang sama. Same set, guys. Same set. Okay. Okay, without further ado, let's start. If you watch my term 1 back then, it was really good because quite a big percentage came out. Sem 1, not bad. But the previous batch even better. I would say the previous batch even better. Anyway, this one here. Okay, so let's start with chapter 7. Guys, I'm doing a brief I'm doing a brief. Okay, can. Okay, I'm doing a brief summary of what you should study first before you study the rest. I didn't say tips. Oh, soalan pertama, objektif ini akan keluar. Oh, this one, question 16, your structure, this will come out. No, those are tips. I'm going through topically what you should focus on. Okay, now, bab 7, guys, I'll use my blue color highlighter. Last year came out bab 7, gut cells. Actual, came out gut cells. Effects of potassium ion uptake. Okay, the previous year don't have. Then oxygen dissociation curve. I think I just colored this up. Bab 7. Enlarge it. Okay, so 2019 oxygen dissociation curve. Hey, how come this one is still the... Hey, this one still haven't changed. I thought I changed it. This one is bab 8. Transport. Bab 7. Bab 7. Still don't have. Then 2017. 2017. You know, 18 cell mediated. Okay, 16 human lung capacity, 2015, 2014. Okay, they can repeat but slightly different, sorry, with a different set of questions. They'll repeat with different set of questions. Okay. So if you ask me, sir, what is important for, what should I study first for bab 7? This is not according to sequence, means not the most important, second most important. No, I'm just telling you what you need to go through first. Okay, guys, although they have asked, you see, whatever came out last year, 90% cannot repeat lah. You can check, guys. You can check yourself. I leave it to you. You check lah. 2021, check with 2020. 2020, you check with 2019. 2019, you check with 2018. I leave it to you to check. There won't be more than 10% similarity. Even the 10% that they repeat, the subtopic, soalan ada perbezaan to maintain that high standard of STPM. Yeah. Okay. So if they want to ask, I would say all these are quite important. Control of breeding mechanism, meaning something to do with chemoreceptors, central peripheral. Then they can ask you CO2 transport. Guys, CO2 transport of carbon dioxide. Oh, 7% dissolve. Then how many percent enter the red blood cell? Carbonic acid, chloride sheath, that CO2 transport. And then, okay, in terms of what they can repeat, guys, they can repeat the gut cells, but they are structure of gut cells. They can ask structure of gut cells, because if you look at this, effect of uptake of potassium ion. If uptake of potassium ion means the process already. Potassium ion enter, osmotic pressure go up, water enter, turgid, stomach open. That one is the process. Okay, what else can they repeat? Sir, can something 2 years ago still come out? Definitely. Definitely. Oxygen dissociation curve. Okay, guys, please take note of this view. Okay, and if you want to, you can also go through this. Very simple, guys. Structure of hemoglobin. Structure of hemoglobin. So these are the few things that you have to pay more attention to before the rest, before all the rest. Okay, control of breathing, CO2 transport, structure of hemoglobin, short and simple. Okay. There are 4 chains, 2 alpha, 2 beta, okay. Affinity towards oxygen. If they want to repeat, can be either one of these. But they might not ask process, they'll ask you structure and go through oxygen dissociation curve. Okay, guys, that's for Bob 7. Okay, let's look at Bob 8. Chapter 8. Chapter 8. Okay, chapter 8 here, I'm going to use another color. Chapter 8 here is transport. It's a long topic. Chapter 8. They asked this last year, formation of first sound. Formation of first sound of the heart, arteriosclerosis, okay, angina. 2021. Then as for 2020, they asked transpirational pool, sugar pressure flow hypothesis, control of heart rhythm. Then as for this one here, the 3 ways of how water moves, cohesion, tension theory, transpirational pool. Okay, high cholesterol, here cohesion, tension theory. Then as for 2014, they didn't ask. Here, arteriosclerosis, myocardial infarction, meaning heart attack. Okay. I still regard it as quite an important topic this year. This year. Okay, so just let me look at my own analysis. It's not easy, you know, do analysis. To even get the questions, many students don't have. Okay, so guys, what is important? What is important for your SEM 2? Now, this topic is divided into animal transport and plant transport. I always emphasize to students, this topic is about animals and then plants. So I'm going to divide it into these two as well. So let's look at in terms of animals, transport in terms of animals, guys, a change in the pressure and volume of the heart. You know, there's a graph. So please take note of that. That one is quite high time already. Guys, that one is quite high time already. Yeah? They can, so how can they ask? They can ask you structure, they relate to cardiac cycle. They relate to cardiac cycle, and maybe they ask you a bit of cardiovascular disease. Means this can be one structure by itself. One structure by itself. Okay? Okay, now, after you have gone through this, first thing, next. Guys, in terms of heartbeat, can you tell me how many types of question are there? Oh, this one, they can really catch you, you know. Guys, essay come out. If they know how to twist sentence, half the candidates can get wrong. Half the candidates can get wrong. Because there are 2 types of mechanism when it comes to heartbeat. Please take note whether they are asking initiation. Initiation, permulaan denyutan jantung. Denyutan jantung means SA node generate electrical impulse spread to the walls of atria. Okay, that is different as compared to, guys, that is different as compared to regulation. The regulation of heartbeat. The regulation. The nervous regulation of heartbeat. Okay, so that one I leave it to you. Okay, now lymphatic system, I normally tell students you either, okay, lymphatic system, you can focus on it either here or chapter 12, but here it's about the same, but that one is more for defense. Yeah, you can read through lymphatic system. Guys, as for Okay, as for plants, there are more things to study because the frequency of how they ask, there's not much you can do lah, guys. Plants, I suggest you to read most of the things. But good news is it's all process. It's all processes, yeah. Cohesion, tension, mass flow. But if you ask me, the three smaller processes have a slightly higher probability simply because they have asked sugar pressure flow here, mass flow. They've already asked this. They've already asked this. Okay, I can see here, guys, cohesion, tension theory. So I would say please read through for both. Please read through for both water and then food transport, but the focus is a little bit more to the three small processes. The three small processes meaning electroosmosis, guys, electroosmosis, cytoplasmic streaming, as well as peristaltic wave. Because the last time this came out was quite a long time ago. Okay. So this is for Bab 8. Guys, this is for Bab 8. Moving on to Bab 9. Okay, moving on to chapter 9, guys. Control and regulation. Control and regulation. Can I have the same color? We go for this purple. I think this purple is quite nice. Okay, sliding filament came out the last semester, the last year actual. Sliding filament came out. I'm not too sure about you guys who are watching only the YouTube, but I dare to say this lah. Okay, maybe I would say most students will find, wow, so lucky it came out. Because last year come out, then this year cannot come out, sliding filament. You might feel so, but I feel very differently because to me, I think I did quite a good job with sliding filament theory with my students. So I asked them in class. I would feel that, I personally would feel that if I'm a student, with the way I learn, sliding filament is a disadvantage that it came out last year. A few of my students really agreed. Of course, guys, you all know lah, tuition, not everyone will respond, about 10%. Mine is considered okay lah, not bad lah, quite active lah. And sometimes they are can I say like not love triangle, it's called like no, scandal is too serious. Okay, something's going on, something's ongoing. Okay, anyway, so to me, it's not a good thing. Sliding filament came out last year. Okay, 2020, hormonal regulation of moth, okay, acidisis, metamorphosis, then plant growth regulator. Plant growth regulator, meaning the hormones are. Okay, guys, before that, resting potential, 2018, hormones, and then acidisis, metamorphosis, auxin, okay, photoperiodism, long day plant, short day plant, guys, curare, 2015, okay, 2014 don't have, then we're able to see potential difference for 2013. Okay, so let's see what is crucial for Bab 9. High time formation. Guys, high time for formation of action potential. Oops, one sec. Next, synapse. Okay, guys, next synapse. Synapse. Okay, synapse. Sliding filament came out last round. They can also ask you to compare. Guys, compare sympathetic and parasympathetic nervous system. Okay, compare sympathetic, parasympathetic. Of course, curare came out, but a long time ago. If you look at 2015, I suggest you go through both cocaine and also curare. Okay, cocaine and also curare. Now, once you are done with this, because cocaine curare is quite short, and if you look at the repeat, if you look at the repeat, non-steroid hormone action came out. Can they still repeat? In my opinion, yes, because no one have the paper. So as you study non-steroid, because it's a very long topic, guys. It's probably the longest topic in the whole SEM 2. As you study non-steroid hormone action, study steroid hormone. And the focus, as usual, is always adrenaline. Okay, the focus is always adrenaline. Okay, as for the steroid, okay, so this is in terms of hormone already lah. So I just cut it here. So the first part is more to nervous system. The second part is more to hormone. Forgot to tell you hormones. So hormones other than steroid, non-steroid, two more things. Number one, phytochrome action. They can still ask phytochrome action, meaning long day plant, short day plant. And one last thing, guys, please go through all the plant hormones. Please go through all the plant hormones. They can ask you, for instance, give 5 functions for auxin, 5 functions for gibberellin. So you have to know the list, guys. You must know the list. All right. So this is for chapter 9. Let me double check. You have to know action potential. You have to know action potential. Resting potential, as you go through resting potential, can read one time. Because it will take about less than 10 minutes. Action potential, resting potential, synapse, compare sympathetic, parasympathetic, cocaine, and curare. Then the hormones, that 3 things, correct? Okay, so guys, these are the things you should really go through first before the other stuff. In fact, I would suggest you go through all the important things, then only the stuff of lesser importance. Okay, bug 10, moving on, bug 10. Another color, let's say purple now. Okay, purple or blue lah, blue. Okay, so anyway, but now chapter 10, reproduction. I use yellow color. Okay, guys, last year, there's nothing to do. Anyway, 10 is a very, I forgot that 10 is a very important topic. This time around, 10 is a very important topic. But let's just go through one thing. So what came out for 10? This one. Oh, this one is labeled wrongly. I thought I changed it. And this 8. The one in your link should be the updated one. Updated meaning the slide topic error have been altered. Okay, so what came out in part 10 last ulangan? The answer is don't have. The previous year was curved, but that one is too far. Okay, so 10 is high time. 10 is high time. Growth pattern came out 2020. Then hormones in early pregnancy. Hormones in early pregnancy, guys. 2017, role of hormone in pregnancy. I'll tell you later why, guys. I'll tell you later why. Growth pattern, embryonic development in dicot seed, 2014, the hormones. Okay, lactation. 2013, parturition. You see this one also have to change. So your updated one should have been improved like this one. Okay, so guys, okay, chapter 10. Chapter 10, spermatogenesis, oogenesis, scan through one time. Now, menstrual cycle, you can go through one time as well. But sir, how come they don't really ask menstrual cycle? Because this is coming from SPM. I always tell students, STPM is not 100% new stuff. There's SPM stuff, then there's the new stuff. The new stuff is is referring to the pregnancy period. Pregnancy period, which means embryonic development. Embryonic development. Okay, embryonic development. Let me look at my analysis. Okay. Even the formation of zygote, embryo, modular blastosis, all those are SPM stuff last time. Okay, so embryonic development is the new content in STPM. So you have to know, of course, read through like menstrual cycle. Guys, embryonic development is important. In terms of two things, the stages, and then the hormones. You get to know the stages and then the hormones. Okay, the hormones during pregnancy. Meaning like these hormones during pregnancy is like what color was it just now? Yellow. This one. This one. Okay. Embryonic development, parturition. Okay, these two are important. Now, again, reproduction, this topic here is about humans and then plants. Then the last part is insects, acidisis, metamorphosis. Acidisis, metamorphosis, like it or not, definitely you have to go through. But guys, for plants, same thing, double fertilization, persenyawaan ganda dua is from SPM. Just scan through one time. Then the important thing is embryo development to form the seed. Okay, seed development. Seed development. Okay, embryo development in the seed, then forming the fruit and all those. Okay, number one. Number two, guys, mobilization of the nutrients in the seed. Make sure you know. If you know this particular process, what's your first point? What's your first point? Imbibition, correct? Water enters the seed. Gibberellin synthesis moves to aleuron layer. Yeah, then function after function. Okay. So human, plants, the last part, no choice, guys. You have to study the whole thing because it's quite short and I think quite okay, not really a problem. Guys, please study the growth curve. and the patterns of growth. This one includes, this is actually the subtopic. This one includes definitely the acidisis and metamorphosis. Have to go through one, guys. This one have to go through acidisis, metamorphosis. So when you study chapter 10, divide it into three components: human, plants, and then the last part. Okay, so tackle the human one, very systematic, then look at the plants one. After that, insects. Okay, moving on. I hope you all can follow. Hope you all still can follow because 11 is homeostasis. I personally like processes, but I know not everyone likes it. Because I find processes is very contained in terms of what they can ask. So guys, another color. This one is like, I think probably I used this now. Can. Okay, chapter 11, homeostasis. Let's see what came out. Okay, enlarge. Let's see what came out for homeostasis. Let's go for this. Or green, go for green. Okay, homeostasis came out last. What happens? Oh yeah, mechanism for sodium ion when a person lose a lot of blood. Then this one is what? This one is the osmoregulation. Osmoregulation. 2020 liver. Yeah, liver. That year I told the students liver can come out, really came out structured. Okay. Urine formation, body temperature, blood osmotic pressure, then liver again. 2016. Oh, this one I didn't color this one. Wait, guys. I just color this quickly. This is 10. This is 10 and 7, never mind. 7, never mind. I was using green. Okay. Chapter 11 here. The adaptation. Okay, and then that's that. Okay, let's look at last year. I mean, sorry, the ulangan 11 don't have. Okay, 11 don't have. Now, guys, I would say this. Top of the list, regulation. This one on top of the list, regulation of pH. Regulation of pH. Yeah. And then please go through ADH aldosterone. Because this is a separate subtopic. I'm sure your teacher told you. Okay. Then adaptation of plants, the last part of the topic. Adaptation of plants. Last one would be this, the urine formation. Then if you got time, just browse through blood glucose, osmoregulation, and body temperature. But these few are top of the list, these few. Okay, chapter 11. Let's see, chapter 11. Because temperature, blood glucose, and osmoregulation are Form 5 stuff. Exactly from, oh, sorry, I missed out one more. It should be here, actually. It should be here. Liver. Because why? Liver and pH is new in STPM. Means you don't find this at all in SPM last time. Okay, but 11. Then pH is quite complex. I took like I took about 45 minutes to really get my students to know it. Then yeah, 45 minutes. Okay, guys, chapter 12. 13 memang you have to go through the whole thing, because 13 is short topic. But I'll still go through with you roughly to show you the frequency. Guys, chapter 12. Okay, let me draw your attention to this. B cells came out last year. B cells came out last year. And for the ulangan B cells and T cells. B cells and T cells. But love it, hate it, guys, love it, hate it, cell-mediated and humoral have to know one. It's always at the very top of the list. Cell-mediated and humoral. But I would say this is good news in a sense. HIV did come out. HIV did come out. HIV did come out. All right. Anyway, 2020, 2019, 2018, cell-mediated, 2017, lymphatic system, 2016, 2015. Okay, then after that, 2014, antibodies. Okay, so top of the list, no matter what, is cell-mediated and humoral, then antibodies. Are you able to explain the structure of antibodies? Are you able to list down the types of antibodies? Where do we find that 5 types? Okay, immunoglobulin G, immunoglobulin N, immunoglobulin A, D, E. All right. And once you are done with this, I think this is quite important this time around. Guys, concept of self and non-self. On our cells, there are a series of antigen. On our cells, there are a series of antigen that allows them to recognize each other. Okay, so that they are considered self. Because our immune system will destroy anything not self. Anything other than this, or anything it doesn't recognize as self, it will destroy. Okay. Oh, sorry. Last one. Last one. SLE. SLE. Okay. Autoimmune SLE. Guys, make sure you know all this. This is quite an important topic. 10 and 12. 10 and 12. Quite an important topic. And last but not least, guys, we have come to the very end of this particular session. I mean, I hope it helps. I hope it helps. Yeah, that's why I purposely come up with this particular video for you. Oops. Oh, and if you are keen to join my class for SEM 3, because SEM 3, where's the guys? Because SEM 3, I'm not sure whether I said this in a YouTube video. The difference between SEM 1, 2, 3 is this. The difference is this. SEM 1 is easy. Of course, if you are my student watching this, you would have heard it many times from SEM 1. SEM 2 is long. SEM 3 is deep. Yeah, the boys especially know very well. Long plus deep. Yeah. Okay. SEM 3, you cannot study by yourself. I mean, you can, but it's very hard. I believe SEM 2, if you study like if you're very hardworking, you are able to memorize most of the stuff, you should be able to answer. But SEM 3 is a different ball game. Shorter, but very much deeper. Again, the boys will get it well. Girls maybe not so much. Short, but very deep. What very deep means you need teachers to guide you. So guys, please listen carefully in school. Please listen carefully in school. This one you really need your teacher to guide you. Your seniors will tell you it's boring. Your seniors means they have finished. Actually, results is out. Yeah. around now. The seniors will tell you it's boring. But I'll use the word dry lah. Very flat, very flat. Oh, let's say, let's say nematoda. But then your syllabus don't have nematoda anymore. They'll give you like one. They give you like one phylum. You need to know all the characteristics. Yeah. Very, very dry. And inheritance is very tough. Inheritance is 3 times tougher than Form 5. I normally take nearly 6 to 8 classes to teach, depending on the performance of that class. Very hard, this one. For STPM level. Yeah. Okay, guys, last part of this session. And thanks for staying until now. Yeah. Okay, bab 13. Bab 13. Now, infectious disease, I don't understand why students don't want to memorize the definition. Guys, can come up one definition? Before flipping to any other pages, make sure you know the definition. Make sure you know the 5 types of infectious agents. Make sure you know the 5 types, guys. If you do not know, go and find out. I don't want to tell you. Go and find out. Okay, and these 4. Sorry. Dengue, malaria, tuberculosis. What's the last one? What's the last one? Yeah, what's the last one? Dengue, malaria, tuberculosis. Let me think. Cholera. Because I was thinking way further already. Okay, now, basically all this here, you have to know the cause. The cause. If there's a vector, like malaria, yeah, like dengue, then the transmission, symptom if got, then what are the roles? It's like what I call BM karangan. Ya, apakah punca, apakah symptom, apakah peranan. Okay, more importantly, let's look at the frequency. Let's look at the frequency, guys. I'll use red color now. So last year they didn't ask, but if I were to, okay, never mind. I check the actual one first. Malaria, I tick 1 tick. Then 2019 don't have. 2018. Now, guys, definition they can ask malaria again. Then 2017 don't have. 2016, 2015. This one, they ask for cholera. I take 1 tick. Okay, 2014. 2000, this one, dengue. Tuberculosis, I thought got 1. Hold on. Tuberculosis, I thought got 1. Don't have. Should have 1. It's a bit small. Malaria. It's supposed to be here. Sorry, because it's very hard to. It's very small the screen. Okay. Tuberculosis. Yeah, I remember got 1. Okay. Now let's look at the in terms of repeat, guys. In terms of repeat, I'm going to use another color to tick. Let's say I use black color. So last year, last paper came out tuberculosis. In fact, I remember tuberculosis got 2 lah. Cholera 1 time. tuberculosis, malaria, yeah, I think that's that lah. I think that's that. Um, let's see, tuberculosis. Okay, yeah. So guys, I think that's that. So please make full use of this handout. How you should also utilize these two pieces of paper is, can you explain sliding filament theory? Can you explain liver? Can you explain blood glucose regulation? If you cannot, please refer to that. You look at the answer or look at your own notes. Prep yourself, guys. Prep yourselves. And last but not least, yeah, so again, back to the advice I told you just now. Active revision. Active revision. Scan through the questions if you don't have time. Look at as many past year as possible, including ulangan, and avoid tough, overly tough questions that doesn't do you any good nearing exam. Okay, nearing your SAM 2. Guys, with that, I wish you good luck. All right. With that, I wish you good luck. For those who are keen to join me in SAM 3, my classes are normally on Mondays. Just in case if there's one student who is keen. My classes on Monday 4 to 5:30. Online only lah. Online only. You can contact my staff. 9399 150. Guys, good luck. Yeah. Take care. Okay.